Good day, everyone. For today's topic, we'll have a thorough discussion on the central dogma of molecular biology. This is the topic outline of this lecture: DNA replication, transcription, and translation. Now, the central dogma of molecular biology was one scientific breakthrough that revolutionized medicine, healthcare, and research. So it's one of the reasons why we can detect viral diseases such as COVID-19 nowadays. It's also one of the reasons why we have more effective treatment modalities right now because of genetic engineering. And genetic engineering is based on the concept of the central dogma of molecular biology, as well as the normal processes that occur in the nature in our nature. It's all because of the central dogma of molecular biology. So, what is the central dogma of molecular biology? So this dogma, when you say dogma, it refers to a principle or a concept or a core concept of a field of study. So the central dogma states that the DNA makes RNA and RNA makes protein. The process by which DNA forms an RNA, that is RNA synthesis, is what we call transcription. And the process by which RNA makes a protein, also known as protein synthesis, is what we call translation. And the process by which DNA is synthesized to form another DNA is what we call replication. Now let's first discuss DNA replication as one of the initial steps in the central dogma of molecular biology. So how does DNA produce multiple copies efficiently? Reproduction is fundamental to all living systems. It's actually one of a characteristic of a living organism: to reproduce, to make copy of itself, to propagate. Regardless of the reproductive mechanism, can be asexual or sexual, a method must exist to transfer genetic material from one generation to the next. DNA must be copied or replicated in a manner that minimizes mistakes, because damage to DNA has has the capacity to be transferred to the next generation if not repaired or prevented that damage. So, one way of propagating our species is through replication. Okay. The one way also of increasing the number of cells is to increase the number of the DNA. Before you make a cell, you first need to make a DNA. So that's one way of DNA replication and ensuring that a cell will have a copy of that DNA. But the process of DNA synthesis is prone to error. So if there are any errors, these errors should be repaired as soon as possible, because if it's not repaired, it is capable of being transferred to the next generation, and it can harm the existence of its offspring. Watson and Crick in 1953 proposed the DNA structure and suggested how it might self-duplicate. According to them, it has not escaped our notice that the specific pairing that we have postulated immediately suggested a possible copying mechanism for the genetic material. So they suggested that replication occurred by gradual double helix strand separation via successive breakage of hydrogen bond, much like the separation of the two halves of a zipper. Since each strand is complementary to each other, each has the information needed to construct the other. Once separated, and each strand can serve as template to direct the formation of the other strand. So according to them, they hypothesized that DNA is replicated in such a manner, just like a zipper. You open up a zipper, and each of the strand of the zipper will form as a template for the production of a new strand of DNA. Now, after a few years, there are several postulations, or theories that tried to describe how DNA is replicated. So the three possible types of DNA replication was the semi-conservative, conservative, and the dispersive replication. For semi-conservative replication, the daughter duplex, this one, the daughter or the products of the replication contains one new and one parent strand. For conservative, the two original strands stay together after serving as templates for the two new strands that also stay together. Therefore, only one contains the only old DNA and the other duplex are new DNA. For dispersive type of replication, the integrity of both parental strand are disrupted. The new duplex strands made up of old and new. So it is a mixture in one strand. It's a mixture of an old and new. Neither the parental strand nor the parental duplex is preserved. So this is an illustration of the semi-conservative replication. As you can see, for each generation, the parental strand is maintained. For conservative replication, the original parental strand is maintained. Both of its strands are maintained or is intact. For dispersive, a certain strand is a mixture of a new and old DNA. Watson and Crick predicted that DNA replication undergoes a semi-conservative nature of replication. So, with this hypothesis, Matthew Meselson and Franklin Stahl in 1957 tested their hypothesis. So what they did is that they grew bacteria in media with ammonium chloride as a sole nitrogen source for many generations of the growth of the bacteria. But I would like to emphasize that the form of nitrogen that they used is nitrogen 15, which is a heavier isotope of nitrogen. The light nitrogen is what we call the nitrogen 14. That's the normal isotope of nitrogen. It's nitrogen 14. But what they did is that they grew bacteria with nitrogen 15 to contain all the bases of the DNA to have heavy nitrogen. They want to remove the N14 and replace it with N15. So basically, that was that 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 was they did. What they did on the initial portion. So first, a washout of ammonium chloride containing nitrogen 15. They put in bacteria with ammonium chloride of nitrogen 14 and removed samples at intervals over several generations and used nitrogen 14 and nitrogen 15 to distinguish between newly synthesized and parental strands respectively. So the purpose of having an N15 and N14 is to distinguish if it's the new strand or the parental strand or the original strand. Okay? If it's the parental strand or if it's the original strand, it's the nitrogen 15. If it's a newly synthesized strand, it's nitrogen 14. So basically, the main point of this study is that they want to test the hypothesis of Watson and Crick. If it's really semi-conservative replication, if one parent strand, if the product of replication contains one parent strand and one new strand, or one old or one new strand. So they extracted the DNA and subjected it to cesium chloride equilibrium density gradient centrifugation to find the buoyant density. And they mix the DNA concentrated cesium chloride solution and centrifuge it until double-stranded DNAs reach equilibrium according to their density. Density of DNA is directly related to the percentage of nitrogen 14 or nitrogen 15 it contains. So Mendelssohn and Stahl, they had several predictions of their experiment. So the prediction of the result of the centrifugation was due to a fact of the three was based on the three postulated methods or modes of replication. So the first prediction is a semi-conservative replication would appear like this. So originally, the original DNA contains all heavy nitrogen-containing nucleobases. So all of the nitrogen, remember that in nucleic acids, we have nitrogen, especially in the nucleobases. So normally it's nitrogen 14, but what they did is that they replaced it with nitrogen 15. So all of the nitrogen of the DNA were nitrogen 15. After one generation of replication, okay, it is predicted that you have a hybrid of nitrogen 15 and nitrogen 14 containing DNA, okay? Because it's semi-conservative. One strand is from the parent and one strand is a newly synthesized strand. Now, these nitrogen 14 came from the environment of the DNA. Then after one generation, half of which contains the hybrid or what we call the intermediate, and half of which contains the light, okay, or the light or the light or the low nitrogen. So when you say light, it refers to nitrogen 14. You say intermediate or hybrid, it's the combination of nitrogen 15 and nitrogen 14. For conservative replication, after one generation, you have a half heavy and half light composition. And after one generation, only 1/4 is heavy and 3/4 is light. For dispersive replication, all are hybrid, all are intermediate after one generation. And after the second generation, all are lighter but not light. So what did Mendelssohn and Stahl observe? They observed that the density of DNA decreases until one generation time when it is halfway between the density of the totally heavy and totally light DNA. So it's a hybrid after one generation, half new and half old. And after two generations have occurred, half of the DNA is totally light and half is hybrid, which contains half light and half heavy, or half containing nitrogen 14, half containing nitrogen 15. While semi-conservative replication continues, the original heavy parental strands remain intact and present in hybrid DNA molecules, but they occupy a smaller and smaller percentage of DNA. So as the generation time increases, the original hybrid um, parental strand is getting lighter. With time, the vast majority of DNA is fully light, with two light strands of DNA. So with this, they concluded that DNA replication occurs in a semi-conservative method or in a semi-conservative way. And that is how they discovered that replication is semi-conservative. Okay, so this is what they observed. So if it was a so these were their prediction. If it was a conservative replication, after the first replication would result into to um, this heavy portion of the nitrogen is located here, while the second one, wherein we have a light band that is present. Okay, for semi-conservative, it's actually in the intermediate or it's in between. The first replication would have a hybrid or the intermediate between the two. Okay, and after succeeding replication, you now have the bands for the light nitrogen. For dispersive, it's just there. It's in the intermediate. And after second replication, the bands are consistently present on the hybrid portion. Okay? So DNA replication is semi-conservative. So each replicated DNA molecule would consist of an old and new strand. So this is the parent DNA double helix. After one generation, that portion of the DNA is the parent. Okay, this portion is the parent, and one strand is a newly synthesized strand. And again, it's the Mendelssohn style experiment that confirmed this process. So we have a sample problem here. Consider the experiment conducted by Mendelssohn and Stahl in which they used nitrogen-14 and nitrogen-15 in cultures of E. coli and equilibrium density gradient centrifugation. Predict the type of replication that would occur given the following results. So you have this following results of the centrifugation. So after one round of replication, you have these bands. You have this band and also this second band. After two rounds of replication, the band is still there. So it's comparable to this one. They have the same hue or the same shade. But after two rounds of replication, the second band is getting darker. And after three rounds of replication, the third the this band or this portion is still darker. Now remember, this portion is the nitrogen 15. This portion is the nitrogen 14 because nitrogen 15 would tend to be at the lower portion because it's heavier. That's why it is called heavy. So this is the nitrogen 15 and this is the nitrogen 14. The intermediate of that would tell us that it is a combination or it is a hybrid of nitrogen 14 and nitrogen 15. Therefore, in this problem, it is a conservative replication. So this mode of replication is the conservative replication. So in this model, the entire molecules serve as a template. After one more round of replication, some molecules will consist entirely of nitrogen 15 and others will consist entirely of nitrogen 14. So two bands should be present. Subsequent rounds of replication will increase the fraction of DNA consisting of entirely new nitrogen 14. Thus, the upper band will get darker. However, the original DNA will still remain as nitrogen 4 and nitrogen 15. So two bands will be present. So an analysis question that I would like you to figure out. In the Mendelssohn style experiment, which of the three modes of replication could be ruled out after one round of replication or after two rounds of replication? Now let's move on to the process of DNA replication. Okay, but I would give the details of DNA replication. I would like to first describe the overall process of DNA replication. So this is the overall process of DNA replication. It starts with the origin of replication. It starts at a certain specific portion of a specific portion of the DNA wherein it is called the orice. Okay, and in this process, first the helicase, which is an enzyme, unwinds the parental double helix. When you say unwinds, it unzips the double helix. Okay. After it unwinds the double helix, molecules of the single strand binding protein or the SSB proteins, they stabilize the unwound template strand. When you say unwound, it refers to the separated strands of DNA. Okay. And after which, the leading strand is synthesized continuously in the 5' to 3' direction by DNA polymerase. Okay, so you have two strands. You have a lagging and leading strand. So later on, we will describe fully about the DNA replication. So the main point here is you have a leading strand and a lagging strand. You have one strand that will proceed in DNA replication continuously in the same direction, in the same direction as the at the same direction of the unwounding of DNA. So if this is the direction, it's towards the left, the leading strand, the direction of the leading strand is also at the same direction as that of the direction of the unwounding or the separation of the replication process. Okay? And one strand is opposite. So the lagging strand would have an opposite direction of DNA synthesis. Okay? Now, again, once the DNA molecules have been separated, the single strand binding proteins would stabilize the template strands, okay, the unwound template strand, to prevent from re-annealing. When you say re-annealing, they would go back together. They would form a double strand together. Okay, so what happens in this portion is that DNA primase or RNA primase begins the synthesis of the RNA primer. Okay, you have a primer before actual DNA is placed. Okay, so in this portion in the lagging strand, we have what we call the Okazaki fragment. Now, this Okazaki fragment is a special type of fragment of nucleotides that only occurs on the lagging strand. Now, DNA polymerase 3 is the enzyme in bacteria that actually places or adds the nucleotides from the primer to synthesize the strand of the DNA. Okay? Now, DNA polymerase 1 removes the primer from the 5' end of the fragment, okay, and it replaces the DNA nucleotides. And once the DNA polymerase removes the RNA primers, the DNA ligase will seal the nicks to complete the replication cycle. So again, in DNA replication, helicase will unwind the DNA. Then the single-stranded binding proteins will stabilize the separated strands to prevent from re-annealing. A primer is placed before actual DNA is laid down. So a primer consists of an RNA, and it is catalyzed, or it is added by this enzyme, a primase. Now, if a primer is already in place, DNA polymerase can now add the DNA nucleotide. Okay, it's DNA nucleotide to the existing primer. Okay? And the direction of replication is from 5' to 3' in direction, meaning from the phosphate end to the hydroxyl end. Again, DNA synthesis occurs from 5' to 3' in direction. Okay? So the enzymes that synthesize DNA cannot initiate the synthesis of a polynucleotide. That is why we need a primer, an RNA primer. We need to have an RNA primer for the replication to proceed. Okay. After an RNA primer is made, DNA polymerase 2 starts to synthesize the leading strand. Okay? And it's the DNA polymerases that adds or catalyzes the synthesis of a new DNA by adding nucleotides to the existing chain. Okay? It adds the first of the nucleotide will bind to the primer. Okay? So the rate of elongation in bacteria is about 500 nucleotides per second. And in humans, it's 50 nucleotides in human cells per second. DNA polymerase, just to emphasize, catalyzes the addition of nucleotide to the 3' end of the growing DNA with the release of 2-phosphate, what we call pyrophosphate. The incorporation of nucleotide into DNA strand, okay, the process of which includes the 3' hydroxyl group at the end of the growing DNA chain, is a nucleophile. Okay, so this is a nucleophile. The hydroxyl group is a nucleophile. It attacks at the phosphorus adjacent to the sugar in the nucleotide, which is then added to the growing chain. And in the process, pyrophosphate is now eliminated and a new phosphodiester bond is formed. So that's the mechanism by which polymerases adds nucleotide to the existing primer. Okay. So what about the lagging strand? So I mentioned that we have a leading strand and lagging strand. When you say leading strand, the direction of replication conforms to the direction of the unwounding of the DNA double helix. So if the replication in the direction of replication is towards the left, then the leading strand would also have a direction towards the left. For the lagging strand, it's opposite. That's an opposite direction. And why is that we have a lagging strand? It's because the synthesis of DNA is always in 5' to 3' in direction. So the synthesis of lagging strand is away from the replication fork. So I forgot to tell you that this portion is what we call the replication fork. This portion that where the DNA is separated. That is what we call the replication fork. DNA polymerase 3 forms the Okazaki fragment in the 5' to 3' direction. Okay, so this is the Okazaki fragment. DNA polymerase 1 replaces the RNA nucleotides of the adjacent primer with DNA nucleotides, while DNA ligase joins the sugar phosphate backbones of all Okazaki fragments into a continuous DNA strand. Okay, so that's the whole overall process of DNA replication. And aside from that, proofreading and DNA repair also occurs. DNA polymerase proofread each nucleotide against its template as soon as it's covalent bonded to the growing strand. Upon finding of an incorrectly paired nucleotide, the polymerase removes the nucleotide and then resumes the synthesis. So a segment of the strand containing the damage is cut out or excised by a DNA cutting enzyme called a nuclease. And the resulting gap is then filled with nucleotides using undamaged strand as a template. So we have several types of nucleases. We have exonucleases and endonucleases. Exonucleases, they degrade DNA from the 3' hydroxy or the 5' phosphate ends, and it works only in linearized DNA. For endonucleases, they break the sugar phosphate backbone of the DNA internally, examples of which are restriction enzyme, the CRISPR-Cas9 system. Now let's have a deeper discussion on the DNA replication. So that's basically the overview. So that you can fully understand about the replication process, we need to see it on the molecular scale. Replication in bacterial cell is actually quite similar with the eukaryotic cell. So we studied DNA replication based on the processes that occur in prokaryotes. Okay? The whole concept is just the same, but in eukaryotes, it's more complex because it involves a lot of proteins. So replication in bacterial cells involves at least 30 proteins needed for the E. coli replication. And several approaches have driven progress in understanding prokaryotic replication. Replication begins at a specific site on the bacterial chromosome. Now remember that the bacterial chromosome is circular. So the site or the specific site in which replication starts to occur is what we call the origin. So it starts at a specific sequence called ORI-C in E. coli on a bacterial chromosome. Again, it's ORI-C. Okay, that is the origin of replication or the start of replication. Many proteins bind at ORI-C to initiate replication. And bacterial replication origin is analogous to the promoter for transcription. So later on, when we describe about transcription, we can also see that ORI-C has a somewhat similar function as that of the promoter region in transcription. Both of them binds specific sequence, specific DNA binding proteins to start process at specific site. Replication moves out from the origin in both direction, in opposite direction, and that is what we call the bidirectional movement of replication. The replication forks, these are specific sites where the pair of replicated segments come together and join the non-replicated segments. Each replication fork corresponds to a site where the parental double helix is going strand separation and nucleotides are being incorporated into the newly synthesized complementary strands. The two replication forks move in opposite directions and meet at the point across the circle from the origin where the replication terminates to form now the newly replicated duplexes, which detaches from one another and go to different cells. So in order for us to understand all of those statements, this is the origin of replication. In E. coli, it is called the ORI-C. Now, during initiation of replication, a replication fork is formed. Okay, and replication may proceed bidirectionally. It can proceed towards this portion and towards this portion. Okay, so that is the usual method by which the prokaryotic DNA is produced. Separation of the strands of a circular helical DNA duplex or a giant linear eukaryotic chromosome poses major topological problems. So the analogy of this one is that you have two-stranded helical ropes and placed in linear piece on the ground. So if you grab both ends, if you grab both strands at one end and begin pulling them apart, okay, it creates this unwinding, right? It creates it creates a tension, okay, at the distant wherein you pull them apart. Okay, strand separation of a double helix also involves the process of unwinding the structure. A rope is free to rotate around its axis, so separation of strands at one end is accompanied by the rotation of the entire fiber as it resists the development of a tension. Okay, so just like this one. So this is the analogy. So you have this DNA double helix. During the process of replication, the strands are separated. Now remember, if you separate the strands, at the distal portion or the distant portion, you create this tension. Okay, so you create a tension or you create this a spatial tightness at the distal portion of the strand. So you need to overcome this tightening at the distant portion. Because if you pull imagine that this is a rope that are bonded together. If you pull them apart, you will tighten this part. So our cells need to have a mechanism to prevent that. Okay. So that is the role of topoisomerase. Topoisomerase can change the state of DNA supercoiling. And in prokaryotes, it's called the DNA gyrase. The DNA gyrase travels along DNA ahead of the fork by removing positive supercoil and changing positively supercoiled DNA into a negatively supercoiled DNA. Now, what happens in DNA replication once the strands are separated, you are actually creating a positive supercoiling ahead of the replication fork. So for example, for this one, this is the replication machinery or the DNA polymerase. At the distant point or at the forward point away from the replication fork, you are actually creating that tension. And that tension is what we call the positive supercoiling. Now, DNA gyrase prevents that one by introducing a negative supercoil. Okay, so it resists the formation of this tension. Okay, what happens is that DNA gyrase cleaves both DNA duplex strands, passing a segment of the DNA through the double-stranded break to the other side, and then seals and cuts. In eukaryotic processes, it possesses similar enzymes that carry out this function. Why is it important to know about this? So we have this drug called ciprofloxacin. Ciprofloxacin is an antibiotic that is usually used in anthrax or even in urinary tract infection, in gonorrhea, in bronchitis. Okay? Ciprofloxacin targets the enzyme DNA gyrase, which is a type of topoisomerase 2. So, uncoiling of the DNA structure is the initiative step for replication, transcription, and repair of DNA. Thus, prolonging inhibition will eventually lead to the death of the cell. So, you're actually targeting the DNA gyrase. So, if you're targeting DNA gyrase, a negative supercoil may not be introduced. So the DNA replication apparatus would have a restriction during replication. So replication process will not proceed because this is already tight. This portion it's very tight. You have no gyrase to counteract that tightness. So in ciprofloxacin, it inhibits that gyrase to prevent the replication of the bacteria, to prevent the replication of the DNA of the bacteria. Now let's move on to DNA polymerase. DNA polymerase synthesizes the new strand of DNA. Now remember again, synthesis of DNA is through semi-conservative replication. Template DNA must meet certain structural requirements to promote deoxynucleotide triphosphate incorporation, or what we call polymerization. So these are the three requirements. You should have an intact, linear, double-stranded DNA that did not stimulate incorporation. You have a single-stranded circular DNA, cannot serve as template, and partially double-stranded DNA works well and yields immediate nucleotide incorporation. So it was soon discovered that single-stranded DNA circle does not serve as DNA polymerase template because the enzyme cannot initiate DNA strand formation. So DNA polymerase cannot initiate, okay? The term here is initiate. They cannot start, they cannot add up nucleotides on their own. So polymerase can only add nucleotides to the 3' hydroxyl end of an existing strand. Therefore, a primer is necessary. So DNA polymerase can only work in a 5' to 3' direction. And all prokaryotic and eukaryotic DNA polymerase have these same two basic requirements: a primer strand to which nucleotides can be added, and a template DNA strand to copy. If in the first place you have no DNA template, what type of nucleotides will now be attached to that specific portion of the DNA? Okay. So this is the process of the addition of nucleotides. So in the process of what we call the polymerization, magnesium ion plays a role. Now I want you to discover what is the role of that magnesium ion in the polymerization of nucleotides during the process of DNA replication that is catalyzed by DNA polymerases. Okay. Now you need to have an existing template in order for your DNA polymerase to work, because that will form as the basis on what nucleotide you will be putting in the existing primer. Now remember, DNA polymerase will only work if there is an existing primer. And our existing primer is not a DNA, but it is an RNA. DNA polymerase findings raise two important questions. Watson and Crick originally predicted that one strand at replication fork would be made in a 5' to 3' direction, while the other one was made in 3' to 5' direction. So how does a 5' to 3' only polymerase do this? So if it cannot initiate strands on its own, how does DNA polymerase initiate synthesis of a new strand in a cell? Okay? So this is the concept of semi-discontinuous replication. Since You have one strand that is 3' to 5', and you have a one strand that's 5' to 3'. Now, in DNA polymerase, the DNA polymerase process, it only synthesizes DNA from the 5' to 3' direction, in the 5' to 3' in direction. Therefore, the template is at the 3' to 5'. Okay? So, in semi-discontinuous replication, all DNA polymerase lay down nucleotides in 5' to 3' direction and move along the template in 3' to 5' direction. So, I will make that clear. In semi-discontinuous replication, the DNA polymerase will lay down nucleotides, meaning they will add up nucleotides in a 5' to 3' direction, meaning from the phosphate to the hydroxyl end, and they move along the original template, the parent strand. They move along in a 3' to 5' direction. Again, polymerase adds up nucleotides from 5' to 3' direction, and it moves through the template in a 3' to 5' direction. Polymerases building both strands move in 3' to 5' direction along template, and both make a chain that grows from its 5' phosphate terminus. One DNA strand grows towards the replication fork, okay, towards the replication fork, and one DNA polymerase will grow in the opposite direction. And again, what is the reason for that? It's because of this original concept. Now, DNA polymerases can only synthesize or can add nucleotides from 5' to 3' direction, meaning it can only move along the template in the 3' to 5'. So in this portion, DNA polymerase will move from this portion towards this portion. For this strand, DNA polymerase will move from this portion, meaning this is the 3' end because this is the 5' end. So this portion towards that portion. So that's the reason why we have those opposing direction. The strand growing towards the fork grows continually in 5' to 3' direction, and that is what we call the leading strand. So again, what's the definition of the leading strand? That leading strand grows towards the fork or towards the site of unwinding of the DNA. The strand growing away from the fork, meaning it runs opposite from the fork, is what we call the lagging strand. And that lagging strand grows continuously as Okazaki fragment. So this portion, okay, these small fragments of newly synthesized DNA in the lagging strand is what we call the Okazaki fragment. Both strands are probably made at the same time, so the leading and lagging terms may not be appropriate as was thought when they were first coined. Strand initiation is done by an enzyme that makes a short RNA primer. Okay? The initiation, again, the start of replication, is catalyzed by an enzyme that creates the primer, and that primer is an RNA molecule, not a DNA. Even if this is DNA replication at the start of the process, RNA plays a role. So RNA polymerase, as a distinct type of RNA polymerase, is found during replication. This is called primase. This primase constructs a short primer made up of RNA, not DNA. Leading strand synthesis is initiated at the replication origin by a primase molecule. Short RNAs made by primase at 5' end of the leading strand and the 5' end of each Okazaki fragment serve as the required primers for the synthesis of DNA by a DNA polymerase. The RNA primers are subsequently removed, and the resulting gap in the strand is filled with DNA and is sealed by DNA ligase. Okay, so just a brief review. As of now, you should be able to describe the Mendelssohn style experiment with regards to the purpose, method, results, and conclusion. should be able to describe the general process of bacterial DNA replication, be able to describe the source and consequences of the torsional stress induced by movement of the replication fork. And in order to counteract the torsional stress, or what we call the tightening of the rope, we have topoisomerase that relieve that stress. DNA polymerase have three major features. They must have a template, they must have a primer, and they must polymerize in the 5' to 3' direction. And you must be able to describe how these features affect the behavior of the replication fork. Now let's continue. Wait. So let me emphasize again. During the initiation of replication, a primer is added before the actual DNA is placed in. So a primer is synthesized by a primase. So you see this color green. These color green are RNA primers. Elongation of the DNA molecule is carried out by DNA polymerase 3. Okay? Primer removal and gap filling by polymerase 1 also occurs. And the presence of the gaps will be sealed by the DNA ligase. Again, to emphasize the leading and lagging strand. So later on, I will show you a video, or I will post a video on the mechanism of DNA replication so that you can easily visualize. How about DNA helicase? DNA helicase is also known as the DNA unwinding enzyme. This unwinds DNA in reaction using energy from ATP hydrolysis to break hydrogen bonds holding two strands together, thus exposing the single-stranded DNA template. E. coli has at least 12 different helicases. DNAB helicase is the product of DNAB gene is the major unwinding machine during replication. It consists of 6 subunits arranged to form a ring-shaped protein that encircles a single strand. It moves in a 5' to 3' direction along lagging strand template and unwinding helix as it proceeds. The DNA unwinding by helicase is aided by attachment of the SSB, or the single-stranded binding proteins to separate the DNA strands. The SSBs bind selectively to single-stranded DNA, or the unwound DNA, to keep it extended and prevent it from being rewound, or to prevent it to come back together. Okay, so the DNA helicase, it is that it separates the DNA molecule, and these single-stranded binding proteins, they prevent the re-annealing or the re-wounding of the separated strands of DNA. Okay, primase enzyme initiates synthesis of each Okazaki fragments. Helicase associates transiently with the primase in bacteria, forming a primosome. Okay, so primosome, it is a complex with primase and helicase. So in the primosome, helicase moves along the lagging strand without being released from the template strand during the lifetime of the replication fork. As helicase moves along the lagging strand template, opening duplex strands, primase periodically binds to helicase and synthesizes short RNA primers that begin the formation of the Okazaki fragments. Primers are subsequently extended as DNA by a DNA polymerase. So once RNA primers are placed, the DNA polymerase will now take its action. Okay. All players of the DNA replication work together in the replication fork. And the replication fork is often to be described as the trombone model. In the trombone model, the DNA polymerase on the lagging strand moves from one completed fragment on the template to a site closer to the replication fork. It hitches with a ride with the DNA polymerase that is moving that way along the leading strand template. The two polymerases are part of the single protein complex, even though they move in opposite direction with respect to each of their templates. Replication of both strands by the two tethered polymerases can be done by having the DNA of the lagging strand loop back on itself so it has the same orientation as the leading strand template. Okay, so this is the usual trombone model. Okay, so have you seen a trombone? A trombone is like it's like a trumpet having a long tube. So I don't have a picture of a trombone, but if you see the on how trombones work, it's similar to the replication fork. So the replication in bacterial cells involves multiple polymerases. We have DNA polymerase 1, 2, and 3. DNA polymerase 1 is mostly involved in DNA repair to correct the damaged DNA section. Consists of a single subunit. It removes the RNA primers at a 5' Okazaki fragment end and replaces the RNA with DNA. DNA polymerase 2 the function of which is yet uncertain, but bacterial mutants have been isolated and have no evident deficiency. DNA polymerase 3 is also known as replicase. It acts in the DNA strand during replication in E. coli. It is a part of a large complex called RNA polymerase 3 holoenzyme or Repliso, which is a large replication machine. DNA polymerases also degrade nucleic acid polymers. Therefore, they have an exonuclease activity. And all bacterial polymerases possesses exonuclease activity. Okay. Exonucleases are divided into 5' to 3' and a 3' to 5' exonuclease, depending on the direction in which strand is degraded. DNA polymerase 1 has both 5' to 3' and 3' to 5' exonuclease activities, along with its polymerase activity. DNA polymerase 1, 5' to 3' exonuclease activity degrades both RNA and DNA, which is unusual since usually exonucleases are specific for and degrade one for the other. Exonuclease removes the RNA primer laid down by the primase at the Okazaki fragment 5' end. Polymerase simultaneously fills in the resulting gap with deoxyribonucleotides. The DNA ligase covalently joins the last deoxyribonucleotide added during RNA primer digestion and the 5' end of the previously synthesized and adjacent DNA fragment. Organisms' survival depends on accurate genome duplication. A mistake in DNA replication results in a permanent mutation in the genetic material and the possible elimination of the cell's progeny or the cell's daughter cells. In E. coli, the chance of incorrect nucleotide incorporation is less than 10 raised to the negative 9, fewer than 1 out of 1 billion nucleotides. The E. coli genome contains around 4 times 10 raised to 6 nucleotide pairs. Thus, this error rate corresponds to less than 1 nucleotide alteration for every 100 replication cycles. Infidelity is detected based on the chemical conformation of nucleotide pairs. Okay. DNA polymerase discriminates among 4 different precursors as they move in and out of the active site. Only one of them forms a proper geometric fit with the template, producing an AT or GC base pair that fits in the enzyme active site. So if there's a base pairing that is incorrect, so for example, for this one, if there's an incorrect base pairing, it can be detected by our DNA polymerase, and they will try to remove that. If incoming nucleotide is correct, a conformational change occurs in which the polymerase rotates, gripping the incoming nucleotides. If the newly formed base pair exhibits improper geometry, the active site cannot achieve the conformation required for catalysis, and the incorrect nucleotide is not incorporated. So the DNA polymerase will also ensure that what you combine with the opposite strand is really the correct nucleotide. If it's A, it should be T. If it's C, it should be G. And if there are any changes for that, it will trigger this improper geometry. The DNA polymerase will somewhat sends a signal that there's a proper geometry pairing between the two. If the base pair exhibits proper geometry, the incoming nucleotide is covalent linked to the end of the growing strand. Despite this mechanism, incorrect pairings sometimes occur around 1 time for every 10 raised to 5 to 10 raised to 6 nucleotides. Polymerase's 3' to 5' exonuclease activity takes care of the mismatched bases. And this can happen right after misincorporation or later on, so what we call a mismatch repair. Thus, there are 3 mechanisms to ensure accurate replication. First, accurate selection of nucleotide. Second is the immediate proofreading. And third is the post-replicative mismatch repair. Now let's move on to the replication in eukaryotes. Now that what we have just discussed a while ago was the process by which replication occurs in E. coli, and E. coli is a prokaryote. Higher organism cells have much more DNA than bacteria and incorporate DNA at much slower rates. Thus, they initiate replication at many sites rather than just one, as with the circular chromosome of E. coli. So for E. coli, you have a circular chromosome. And you have the origin of replication occurs at one site called the oric. But in eukaryotes, we have multiple origin sites because of a very, very long or very of a very long DNA. Initiation of DNA synthesis in a given cell is subject to replication, especially in eukaryotes. Replicons close to each other on a given chromosome tend to replicate simultaneously. And in mammals, timing of replication in a chromosomal region may be determined primarily by the action of genes in the region and or its states of compaction. The less active, the more tightly compacted the DNA. The later is the stage at which it is replicated. The replication of eukaryotes is also complex. Events occurring at the replication forks are very similar to prokaryotes regardless of the genome. Okay, but the proteins are usually complex. Okay, so for E. coli protein, these are the proteins required for replication. For E. coli protein, you have list of this one, and in eukaryotic protein, we have a list for this one, and also their following functions. Okay, so instead of a SSB, you have the RPA protein, which has a similar function in the single-stranded binding proteins. But generally, the process is still the same. Okay, it's just that several proteins are added. So by now, you should know the functions of DNA polymerases, the helicase, the primase, the primosome, okay, and the DNA ligase. You should be able to demonstrate how and why the trombone model is used to describe the DNA replication. Exonuclease activity can correct mistakes in DNA replication, and you should be able to describe the important differences between eukaryotic and prokaryotic replication. You should be able to name eukaryotic or prokaryotic replication enzymes and their function. So in eukaryotes, we have also multiple polymerases, just like in the prokaryotes. And eukaryotes have at least 5 different DNA polymerases. You have the alpha, beta, gamma, delta, and epsilon. 3 of them are involved in nuclear replication, 2 of them are not. All elongate DNA in a 5' to 3' direction, and none of them is able to initiate the synthesis of a DNA chain without a primer. So just like in prokaryotes, you need to have a primer. You need to have an RNA primer to be able to synthesize DNA chain. Polymerases gamma, delta, and epsilon possesses a 3' to 5' exonucleases. Polymerase gamma replicates the mitochondrial DNA, and it is not involved in nuclear DNA replication. Polymerase beta functions in DNA repair, and it is not involved in nuclear DNA replication. For polymerase alpha, it is tightly bound to primase. Together, they initiate the synthesis of each Okazaki fragment, and primase lays down a short primer, and then polymerase alpha extends with 20 deoxyribonucleotides. Polymerase delta assembles leading strand and most of the lagging strand fragments. It's thought to be the primary replicative enzymes, and it requires the sliding clamp structure, just like the polymerase 3 in E. coli. Polymerase epsilon determines determining its role has been difficult, and replication cannot be finished in cells lacking this polymerase. So you need to have a polymerase epsilon in order for replication to occur. In eukaryotes, we have what we call the nuclear organization. Now, unlike in prokaryotes have no nucleus, and their chromosomes, their chromatins and chromosomes are somewhat very different from the eukaryotic chromosomes. Replication apparatus consists of a huge complex of proteins operating within the confines of the structured nucleus. Increasing evidence suggests that replication machinery is present in tight association with the nuclear matrix, and replicating DNA moves like a conveyor belt through the immobilized replication apparatus, rather than remaining stationary. Nucleosomes form on both daughter duplexes very near in the replication fork. This indicates that assembly of DNA into nucleosomes is a very rapid event. And in vitro studies indicate that the H3H4 tetramer present prior to their replication remain intact and are distributed randomly between two daughter duplexes. The two H2A and H2B dimers of each parental nucleosome do not remain together as the replication fork moves through the chromatin. Okay? So DNA is very susceptible to environmental damage. We have types of damage experienced by DNA. These are ionizing radiation, exposure to a variety to reactive chemicals that can alter DNA bases, ultraviolet radiation, and thermal energy. For ionizing radiation, it can actually break the DNA backbone. Exposure to chemicals can alter the DNA bases. Ultraviolet radiation causes adjacent pyrimidine, such as cytosine or thymine, to interact covalently. This is sometimes what we call the thymine dimers. Thermal energy generated by metabolism in a warm-blooded bird or mammal can split adenine and guanine from their attachment to DNA backbone sugars. And spontaneous alteration or lesions often occur. Each cell of a warm-blooded mammals loses around 10,000 bases per day. So what are the potential effects of DNA damage? If the DNA damage occurs in the gametes, when you say gametes, it's the sex cells. These are the cells that are involved in the production of your sperm and egg. So if damage occur in a cell destined to become a gamete, the damage or mutation can be passed on and become a permanent part of the population's gene pool. If it's from a somatic cell, when you say somatic cell, it's from a body cell that is not involved in fertilization in sexual reproduction. It is not possible to pass the mutation on, but can interfere with replication and transcription, can lead to malignant transformation of a cell. So this can result to cancers, and they can speed the process by which an organism ages, and it is vital that cells have a way to repair the damages they do. Now, DNA repair mechanism can be carried out by our cells through the nucleotide excision repair. This is one method. In nucleotide excision repair, it removes part of strands having lesions, so having the lesions of pyrimidine dimers and chemically altered nucleotides. It has a mechanism called cut and patch. Transcription coupled pathway is one pathway of nucleotide excision repair in which template strands of genes that are being actively transcribed are preferentially repaired. Okay. Repair of template strand is thought to occur as DNA is being transcribed. The presence of the lesions may be signaled by a stalled RNA polymerase, and this ensures that those genes of greatest importance to the cell, the genes being actively transcribed, and they receive the highest priority on the repair list. For global pathway, they usually have a slower, less efficient pathway that corrects DNA strand in the remainder of the genome. What are the steps for the nucleotide excision repair in eukaryotic cells? First, the lesion is recognized by protein scanning DNA. It recognizes distortion sites in helix. The recruitment of repair enzyme to the lesion is the next step. The damaged strand is cut on both sides of the lesion by a pair of endonucleases. So the problematic, for example, if you have this thymine dimers are usually formed from um, ultraviolet light. So this is also one way of, or this is also the basis of the UV light. So it creates this pyrimidine dimers, or what we call thymine dimers. So it forms a covalent bond with adjacent thymine dimer. And this is a type of damage. So your cells will try to detect that. So again, lesions can be recognized by our repair mechanisms. That damaged portion will be cut by endonucleases. Okay, and a segment of DNA is released, and the gap that you form from the cutting of this portion of the DNA is filled by DNA polymerase. And the nicks that are present will be sealed by a DNA ligase. Now we have also this type of mechanism, the base excision repair. Sometimes single nucleotides in the double helix are altered via chemical reaction and become mismatched. For uracil, it forms by hydrolytic removal of cytosine's amino group. So if you remove amino group from cytosine, you form uracil. 8-oxoguanine is caused by damage from oxygen-free radical, and 3-methyladenine is caused by alkylating agents. It is the transfer of methyl group from a methyl donor. So in base excision repair, is initiated by a DNA glycosylase that recognizes the alteration. For example, in eukaryotes, you, you typically don't have a thymine in the DNA. Now, once the amino group of cytosine is removed, you now form uracil. So this uracil is an abnormal mutation. So DNA glycosylase recognizes this alteration. And aside from um, detection, it removes the base, but not the entire nucleotide. Unlike in nucleotide excision repair, the entire portion of the nucleotides wherein you have the problem of the thymine dimers are cut using an endonucleases. But for glycosylase, only the bases are removed. So what are the steps of base excision repair in eukaryotes? First is the beheaded deoxyribose phosphate is removed by endonuclease and DNA polymerase. The AP endonuclease cleaves the DNA backbone, and polymerase beta removes the sugar phosphate remnant that has been attached to the excised base. And the gaps are filled by DNA polymerase beta, and the strand is sealed by DNA ligase. So this is the mechanism by which base excision repair occurs in eukaryotes. Another portion is the mismatch repair. For mismatch repair, cells replace mismatched bases that are incorporated by DNA polymerase and escape the enzymes proofreading nucleases. Mismatched base pairs causes distortion in double helix geometry that are recognized by repair enzymes. So this is one mechanism, mismatch repair, to detect the mismatch bases. So we won't be discussing thoroughly about mismatch repair because it's quite complicated. So with all of that, I would like to present the complex issues in DNA replication. So these are the common physiologic issues that we encounter in DNA replication that our cells need to overcome. Number one, the helix must undergo localized unwinding, and the resulting open configuration must be stabilized so that synthesis may proceed along both strands. And this complex issue, or this issue, is um, is taken care of by the single-strand binding proteins. As unwinding and subsequent DNA is synthesis proceeded, increasing coiling creates tension further down the helix, which must be reduced. And this problem is addressed by the DNA topoisomerase or the DNA gyrase for bacteria. A primer of some sort must be synthesized so that polymerization can commence under the direction of DNA polymerase II. And surprisingly, RNA and not DNA serve as a primer because our DNA polymerase cannot synthesize on their own, needs to have that 3' hydroxyl group from a primer. So you need to have a primer. And this is problem is addressed by the primase enzyme. Once the RNA primers have been synthesized, DNA polymerase begins to synthesize the DNA complement of both strands of the parent molecule. Because the two strands are anti-parallel to one another, continuous synthesis in the direction that the replication forks moves is possible along only one of the two strands. And on the other strands, synthesis must be discontinuous and thus involves somewhat different process. Okay, because of you have a different polarity of the two strands, you now have the leading strand and the lagging strand. And in the lagging strand, you can find the Okazaki fragments. The RNA primers must be removed prior to the completion of replication, and the gaps that are temporarily created must be filled with DNA complementary to the template at each location. So the removal of primers are done by DNA polymerase 1, and the gaps are addressed by the DNA ligase. Okay. The newly synthesized DNA strand that fills each temporary gap must be joined together with the adjacent strand of DNA that's called the DNA ligase. And when DNA polymerases accurately insert complementary base during replication, they are not perfect, and occasionally incorrect nucleotides are added to the growing strand. So a proofreading mechanism that also corrects errors is an integral process during DNA synthesis. And that is why we have several DNA polymerases that have proofreading mechanism and exonucleases activity to remove those abnormal or incorrect nucleotides or nucleobases that are added during the process. So to end our discussion, I'm giving you an assignment for you to have a comprehension check. So study again the slides, supplement your study with our references. Okay, and I actually recommend I don't recommend any a specific book, as long as it's biochemistry or genetics book. You can actually read on it. Okay, and you can also find several videos in YouTube help you that can help you to visualize what is happening during the replication process. So for your assignment, number one is to list all the proteins and enzymes used in or involved in DNA replication in prokaryotes and describe their molecular function. Number two is to ask the question, why is there a leading strand and a lagging strand? Number three, why is the synthesis of DNA runs from a 3' to 5' in direction? Number four, why is there a need for an RNA primer in DNA replication? Why do you need to have a primer? If it's a DNA replication, why is an RNA involved? Fifth, how does repair system know which member of a mismatch repair is the incorrect nucleotide? And sixth, what are the similarities and differences of eukaryotic and prokaryotic DNA replication? Now, I keep on loving these types of question because I like to ask questions that will involve comparison and contrasting. When you say comparison, that is to find the similarity. Contrasting is the differences. So try to see the components at its detail so that you can get a bigger picture of what is really happening during the replication. Okay. You need to compare the enzymes, compare the prokaryotes and eukaryotes, you compare the leading strand and lagging strand. Okay. You also compare the other processes of the central dogma, such as transcription and translation. How are they similar to each other, and how are they different from each other? Okay, that's one way for you to understand the whole process of the central dogma of molecular biology. So that actually ends my first part of the lecture on DNA replication, and please see the next video for the transcription and translation. Thank you so much and have a great day.